Study Section 3. Introduction. By means of different patterns of synaptic connections between neurons, synaptic circuits are constructed during development to carry out the different functional operations of the nervous system. The human brain contains about 100 million neurons, and the average neurons form something on the order of 1,000 synapses. Then there are stars in our galaxy. Kandel and Seegerbaum, 1995. This lecture provides a better understanding of the synaptic transmission. Objectives. At the end of this lecture, you should be able to 1. Explain synapse. 2. Identify the forms of synapse. 3. Explain what is meant by glial cells. And 4. Describe neurotransmitters. Contents. The synapse. Neurons do not come into direct contact with one another. They are separated by tiny gaps called synapse. The synapse is the smallest and most fundamental information processing unit in the nervous system. Synapse takes one or two forms of electrical synapse or chemical synapse. Electrical synapse directly stimulates adjacent cells. Chemical synapse stimulates adjacent cells through the use of chemical messengers. Freeberg, 2006. Diagram of anatomy of a typical synapse. Electrical synapses. Electrical synapses between two cells are not only found in animals, but in plants as well. And this suggests that they are very ancient in terms of evolution. The gap between an electrical synapse is quite small. It is only 3.5 mm wide. In contrast, the average gap between presynaptic neurons and postsynaptic neurons at a chemical synapse is about 20 mm wide. The presynaptic and the postsynaptic cells at an electric synapse are joined by special protein channels that essentially connect the two cells. These channels make it possible for positive currents from the neuron presynaptic to flow directly into the postsynaptic neurons. The main advantage of the electrical synapse is speed. Transmission of a message from one cell to another is nearly instantaneous. In contrast, chemical synapse takes between 0.3 mm to several milliseconds in order to complete the series of steps involved with transmitting the message from one cell to the next. Although electrical synapses are fast, chemical synapses have the advantage of providing a much greater variety of responses. The only type of message that can be sent at an electrical synapse is an excitatory one. Excitation means that only one cell can tell the next cell to produce an action potential. Another major advantage of chemical synapses over electrical synapses is that a very small presynaptic neuron using a chemical messenger can still influence a large postsynaptic neuron. In the case of an electrical synapse, it takes a very large presynaptic neuron to influence a tiny postsynaptic neuron. And this is simply due to the electrical requirements of this type of transmission. Chemical synapses. In chemical synapse, neurotransmitter substance released by the presynaptic neuron floats across the synaptic gap to receptor molecules embedded in the synaptic membrane of the postsynaptic cell. Although this takes more time than transmission at an electrical synapse, it allows neurons to send a much variety of information. The argument that neurons communicate with each other via a chemical transmission process is therefore robust. Discussion of signaling at chemical synapse involves two steps. The first step is the release of neurotransmitter chemicals by the synaptic cell. The second step is the reaction of the postsynaptic cell to the transmitters. The second step is the reaction of the postsynaptic cell to the neurotransmitters. Chemical synapses are found only in between nerve cells or between nerve cells and the gland cells and the muscle cells that they innervate. The postsynaptic membrane contains receptors from the transmitter substance released from the presynaptic terminal. Prior to release, molecules of neurotransmitters are stored in synaptic vesicles. The vesicles contain substances involved in the chemical transmission process of the neurons called neurotransmitters. The process by which the vesicles release their content is known as exocytosis. After its release, the transmitter acts on a receptor in the postsynaptic membrane. There is also a deactivation of the neurotransmitter. The neurotransmitter is either destroyed or taken back into the terminal from which it came, where it can be reused or degraded and removed. Neurotransmitters. Neurotransmitters are specialized chemicals that carry neural information. Neurotransmitters act on neurons in their own immediate vicinity, generally at a synapse. A neurotransmitter can be thought of as a key and a receptor as a lock. The same type of key can in principle be used to open many different types of locks. Receptors can be classified broadly as excitatory, causing an increase in firing rates, inhibitory, causing a decrease in firing rates, or modulatory, causing long-lasting effects not directly related to firing rates. In principle, a single neuron releasing a single neurotransmitter can have excitatory effects on some targets while having inhibitory effects on others. Some of the major transmitters substances that have been identified include non-epinephrine, NE, also known as non-adrenaline, 5-hydroxytryptamine, 5-HT, also known as serotonin, dopamine, DA, gamma amino butyric acid, GAMA, acetylcholine, ACH, and are used excitatory neurons to stimulate postsynaptic neurons, while others such as GABA have inhibitory effects. Nguru Okorie, 1990. The tiny space that separates the presynaptic space or synaptic cleft contains the extracellular fluid that provides a medium through which the neurotransmitter substances can diffuse to the postsynaptic membrane. When an axon fires or propagates an action potential, a number of synaptic vesicles migrate to the presynaptic. They adhere to the membrane and rupture. The rupture spews their contents into the synaptic cleft, thus releasing the chemicals into the gap. Sohnen and Rothman, 1994. Shearer and Nichols, 1993. Following this, the chemicals diffuse through the synaptic fluid to reach the postsynaptic membrane. After the nerve impulse has crossed the synaptic gap and the impulse transmitted to the postsynaptic neurons, the neurotransmitters are destroyed by an enzyme or reabsorbed into the presynaptic vesicles. Smith, 1998. This process moderates the message transmission function of the neuron. The valuable and useful message transmission function of the neuron is further highlighted in the next section, which discusses its parent structure, the nervous system. other cells and structures of the nervous system. We have discussed characteristics of neurons and how they communicate. It must be noted that neurons are not only the cell making up the brain. There are other non-neural cells. Even though neurons are the most important, the cellular machinery of the brain also depends on the active participation of the glia, vascular elements, and the connective tissue elements. Myelation of PNS and CNS axons. Glia. The extracellular space between the nerve cells is fixed with specialized support cells called glia. Unlike the neurons, glia does have the capacity to divide and form new cells. The glia, whose name comes from the Greek word for glue, serves a variety of support functions for neurons. In the human nervous system, there are at least 10 to 50 glia for every neurons. Freeberg, 2006. Glia are generally categorized by size. The microglia are the largest varieties of glia cells, while the microglia are the smaller varieties. There are three primary types of microglia, which are astrocytes, oligodendrites, and Schwann cells. The astrocytes provide a variety of support functions to neurons, whereas the oligodendrites and Schwann cells supply the myelin covering that insulate axon fibers. Astrocytes derive their name from their star-like shape. Astrocytes are the most common type of glia in the brain. Astrocytes are thought to clean up excess transmitters and ions from the extracellular spaces, thereby helping to keep the interactions that take place on the neuron surface free of background chemical noise. Astrocytes may also contribute glucose to very active nerve cells, and they may redirect the flow of blood and therefore the transport of oxygen to especially active regions. This glia may also be important in providing some of the signal essential for the regulation of synaptic function. Although none of this proposal is yet certain, scientists do know that after minor damage to the brain, astrocytes scavenge the dying neurons, an action that helps limit the spread of toxic substances. Bloom et al. 2001. Oligodendrite type of glia is better defined by their function than by their shape. Some axons are insulated in a way that customizes them for rapid conduction of electrical impulses. This cellular insulation, called myelin, is a compact wrapping material consisting of layers of membrane that project from the oligodendrite cell body. Oligodendrites and Schwann cells provide the myelin covering that insulates some axons. Peters, Parkey, and Webster, 1991. Oligodendrites provides the myelin in the central nervous system. Schwann cells supply the myelin for the peripheral nerves, exciting the brain and spinal cord. A single oligodendrite puts out a number of processes that wrap themselves around the axon of adjacent neurons. Because a single oligodendrite may myelinate axons from an average of 15 different neurons. This microglia contributes to the stability of the brain and the spinal cord. Schwann cells. In the peripheral nervous system, the glia cells that form myelin is known as the Schwann cells and have slightly different synthesizing abilities and chemical properties from those of the oligodendrites. A single Schwann cells provide myelin for only one potential axon. It takes a large number of Schwann cells to myelinate a peripheral nerve. Freeberg, 2006. We can see the importance of myelin by studying conditions in which it is damaged. The diseases multiple sclerosis, MS, involves a progressive demyelination of the nervous system. The end result is neural signaling that may not work properly, leading to a range of symptoms from mild increased fatigue to more severe mobility problems to death. Oligodendrites and Schwann cells differs in their reaction to injury. The Schwann cells actually help to guide the regrowth of damaged axons, whereas the oligodendrites lack this capacity. Microglia. You may not like the idea of having brain debris. However, like any other cell, neurons and glia do die, rather than leave the debris lying around where it might interfere with neural functions. The microglia serves as the brain cleanup crew, like certain astrocytes. These tiny cells are mobile and can travel to locations of injury where they digest the debris. Vascular elements. Among the other neuronal cells of the nervous systems are those of the blood vessels, like the arteries, veins, and capillaries, which make an essential contribution to its vitality. The brain receives a privileged share of oxygenated blood. In fact, all the muscles of your body together, when fully active, draw only about 25% more oxygen than the brain does. The blood vessels of the central nervous system are unlike those of the rest of the body in that they lack the ability to transport large molecules across their walls. Connective tissue elements. These non-neuronal cells line the outer and inner surfaces of the central nervous system. Within the bony continues of the surfaces of the skull and the spinal cord, the CNS is sealed into a form-fitting, fluid-filled stocking of membranes called the meninges, made up of conventional connective tissue found elsewhere in the body. The fluid is the cerebrospinal fluids. The meninges and cerebrospinal fluids act as a shock absorber system to soak up the twists, turns, bumps, and other insults of the body that will severely hamper the integrity of the nervous system, where they are to be transmitted full force. Connective tissue elements. These non-neuronal cells line the outer and inner surfaces of the central nervous system. Within the bony continuums of the surfaces of the skull and the spinal cord, the CNS is sealed into a form-fitting, fluid-filled stockings of membranes called the meninges, made up of conventional connective tissues found elsewhere in the body. The fluid is the cerebrospinal fluid. The meninges and the cerebrospinal fluid act as a shock absorber system to soak up the twists, turns, bumps, and other insults to the body that will severely hamper the integrity of the nervous system, where they are to be transmitted full force. Summary. 1. The synapse is the smallest and most fundamental information processing unit in the nervous system. 2. Synapse takes on or two forms: electrical synapse or chemical synapse. 3. The gap between an electrical synapse is quite small. It is only 3.5 mm wide. 4. In contrast, the average gap between presynaptic neuron and postsynaptic neurons at the chemical synapse is about 20 mm wide. 5. In chemical synapse, neurotransmitter substance released by the presynaptic neuron floats across the synaptic gap to receptor molecules embedded in the synaptic membrane of the postsynaptic cells. 6. Neurotransmitters are specialized chemicals that carry neural information. 7. The extracellular space between the nerve cells is filled with specialized support cells called glia. 8. Glia are generally categorized by size. The microglia are the largest, are the smaller varieties. 9. There are 3 primary types of microglia, which are astrocytes, oligodendrites, and Schwann cells. 10. In the peripheral nervous system, glia cells that form myelin is known as the Schwann cells. End of study section 3. Thanks for listening.